This next presentation is going to be given by Dr. Phil Young from the Down East Institute, and he's going to talk about larval transport and connectivity among mussel populations.
Okay, thanks, Paul. I'm going to try to go without the mic. Let me know if you can't hear me in the back. So for the last couple of years, we've been sort of wrestling with this problem of, we're working with mussels. Where did the seed come from? Where do they go in eastern Maine? Go ahead. And I want to point out, I'm the guy up here talking, but this is the work of a whole bunch of people. So these are all my co-authors on this who've been involved in various parts, and I've tried to stick their names on the appropriate parts. So marine ecologists consider this problem one of population connectivity. So that's the label we put on it. So why bother studying population connectivity? Well, shellfish, as you know, probably know, have a larval stage that can potentially move very long distances. That means the adults may live and grow up a long way from where they were conceived. And the movement of larvae then interconnects populations. And understanding these connections then becomes critical for a lot of management applications. First, if you're harvesting in one area and want to understand how that's going to impact the seed supply to another, you need to know where the seed are going. Similarly, if you wanted to create, say, a reserve that was going to produce seed that would get to other areas, you want to do that in an area that's a net exporter rather than a net importer. And in addition to sort of those direct management applications, if we sort of understand more about this process, then hopefully it may help us explain more about variation in recruitment, though that's not a guarantee. Next, please. And just to point out that we have a recruitment problem with mussels. So this is a data set for Swans Island, not a very big population, but as you see, a very steady decline in recruitment over the last 10 to 15 years. And I think most people would agree that that's sort of representative of what's happening in most of eastern Maine. Next, please. All right, so when we start worrying about this problem of how's the seed moving around, in eastern Maine, the sort of dominant feature we need to worry about is we have this really strong current that runs down the coast. The eastern Maine coastal current. On ebbing tides, the current speeds out there can exceed 1 meter a second, very strong. Originates from the Bay of Fundy and from sort of over here off of Nova Scotia. Next, please. So the first possibility that kind of leaps to mind in terms of supply relationships is maybe eastern Maine is on sort of the downstream end of the supply coming from Canada. Maybe all of our seed are coming out of the Bay of Fundy or from Nova Scotia and making it into the bays over here. Next, please. On sort of the extreme opposite end of that, another possibility is that the larvae that are produced in these bays never escape from the bays. And the bays are basically seeding themselves. If that were the case, it would be wonderful from a management perspective because it would mean you could just manage them all independently. And this would be likely to happen if there's not much exchange between the water in the bays and that strong coastal current. Next, please. And sort of as an intermediate possibility is maybe the larvae are getting out of the bays, but all they're doing is moving down a few bays over to neighboring bays. And presumably, if this is going on, most of the movement is sort of from the northeast to the southwest, because that's, again, the direction of the prevailing current. Next, please. But it's also possible that we could have some, at least more limited exchange moving back to the northeast. And this would happen if you've got sort of localized flow fields inside of this coastal current that bring larvae in those directions. All right, so we've been trying to sort out those different possibilities. Next, please. And the sort of occurrence of those three different pathways ultimately really depends on this question of the extent to which the water in that coastal current is mixing with the water in the bays, which oceanographers would refer to as a cross-shelf mixing. That's the label I'll stick on it. Next, please. And just to sort of give you a concrete example of why this matters, this is output from a model modeling larval lobster dispersal from Nova Scotia. And the color coding just represents where the model larvae are X number of days after they were released. And these folks really didn't know how much a cross-shelf mixing, how much mixing there is between that coastal current and the bays. So they just modeled different scenarios, okay? This is the one where there's more mixing, this is the one where there's less mixing. And when you look at these two figures, the first thing that grabs you is, gee, on a large scale, it doesn't really matter much. If you're sitting down here and you find lobster larvae in the water, the probability that they came from Nova Scotia is about the same under either scenario. Next, please. But where it really starts to matter is if you're a harvester or a resource manager sitting in one of these bays along the coast, because in this scenario, you get those larvae arriving in your bays, and in this scenario, even though there's plenty of larvae out there, they just go scooting on down the coast and they don't get into your bay. So with that as sort of an introduction, I'm going to try to very quickly talk about 6 things today. I'm going to start out just presenting some physical evidence of how much mixing there is between this coastal current and the bays in eastern Maine. Look at the ramifications of that for how the sea mussels are distributed in the water across the shelf off of Pleasant Bay. And then from here I'm going to sort of take what is going to appear to be a detour and start talking about some of the data we've been collecting in the actual mussel beds. And the reason for this is in order to, ultimately what we want to do is model this process. In order to do that, we need to know what the seed output of the mussel beds is, and we need to know something about the timing of spawning and settlement, when are the seed in the water. Next, please. And I think you'll see as I go through these that these actually turn out to be pretty interesting in themselves, though ultimately our original motivation was to provide data for step 5. And then finally, I'll finish up by telling you a little bit about using natural tags to sort of empirically assess where the larvae move to. Next, please. And the first 4 of these are relatively finished products. These last 2 are kind of work in progress, so we're not going to have all the answers. All right, so first just asking how much physical mixing occurs between that coastal current and the bays. When we first got interested in this problem, we explored that by deploying what are called drifters. And a drifter is just a real simple little structure made out of PVC and vinyl. It's got lots of surface area, so it's grabbed by the water, so it moves with the water currents. It hardly sticks out of the water at all, so it doesn't get blown around by the wind. And on the top here, it has a satellite transmitter. So you just turn these things loose, and they phone home and tell you where they are over time. And so when we release a bunch of those in the Grand Channel, you see up here in the channel, there's an awful lot of mixing going around. They're going in circles, they run aground on the shore. There's no separation between this coastal current and the shore up in the channel. Then there's sort of this transition zone from, say, Machias Bay down to Chandler, where there's still some mixing. But beyond that, once they exit that, they basically just head straight to the southwest. And you notice they don't go into any of those bays along there. So lots of mixing to shore, maybe episodic, limited mixing, and then this very big disconnect between the bay waters and the coastal current down here. Next, please. Similarly, there's a longer-term drifter set that Noah's put together. Here we just looked at the paths of drifters. They weren't released in this area, but they just happened to come through it during June and July, during the time period of interest. And you see they pretty much show the same pattern. One of them came in and sort of explored the lower reaches of Frenchman Bay. That's about the furthest anyone got. Please. So, all right, given that we have this kind of disconnect, we got this coastal current flowing along the coast, there's not much mixing between that and the bays, what would the consequences for that be for how the mussel seed are distributed across the shelf? We've explored this question off of Pleasant Bay. First, we know there are mussel beds in here, so we expect to find mussel larvae on this end of the transect. Similarly, we know there's lots of mussels in Canada and Cobscoat Bay, so there probably are larvae out here in this coastal current. And if they're not mixing across the shelf there, we expect to also have them be abundant out here. But what we don't expect is to see them homogeneously distributed across the coast, and we might see very different temporal patterns on the 2 ends of that transit. Next, please. So this is the summary of a lot of work, and it's a little bit complex, but what we're plotting here, on the y-axis, these are the inshore stations moving further offshore, time on the x-axis, and these are sample dates that are 2 weeks apart. The color background here is the water temperature at the 5-meter depth where the larvae were sampled, and you see it's basically warming up as the season goes on, and you're developing a bigger gradient sort of across the shelf there. And the actual data on the mussel larvae are these circles, and the size of the circle is proportional to the number of mussel seed. And so the pattern you notice is first, they show up on the inshore and a good 8 weeks before we ever see them offshore. They're quite abundant 4 weeks before they first appear offshore. When they are abundant, you see sort of a gradient across the shell, okay? But you never see, you see it looks like there's a batch of larvae in here and they were diluted moving out, but they didn't make it all the way out to the EMCC. Sometimes you find them abundant at both ends, but there's always at least one site where there aren't very many of them in between. Next, please. And so when you sort of put all of that together, what it sort of suggests is that we've really got two different groups or populations of larvae, and that there's an inshore produced group that are staying more or less inshore. We do have Canadian larvae out there, but they're way offshore, and they're just zipping by the bays. These two groups probably intermingle somewhat in here, and I don't know exactly what the borders are, but it looks like they're not intermingling to the extent that these larvae would be making it in the shore. Next, please. All right, so I'm going to briefly leave the oceanography for a minute and turn to some of the data we've been collecting in the actual mussel beds, and then I will sort of, via this, sort of slowly work my way back to the oceanography. So first of all, for those of you who do not live or work in eastern Maine, I need to educate you about mussels, because you probably think that mussels live on the rocks and on exposed shores, and in eastern Maine they do not. In eastern Maine, they're basically absent on exposed rocky shores. And what they do is they form huge beds up in the estuaries. And they're basically either on top of the clams, on the clam flats, or they're within about a quarter of a mile or so of the clam flats. A lot of these beds are intertidal. There are some subtidal ones. And they really are in pretty soft setting. So this is the set of populations that we've been monitoring and working with. Next, please. We sort of define our study areas within this ellipse here, where we do get some data on one sort of upstream reference population. Next, please. And one of these we don't actually have data yet. This is the Jordan River bed we haven't gotten to yet. Next, please. And a couple of these are really minor, not of any commercial interest. One is right at our institution, so it's easy to monitor. And we wanted something in Machias Bay. But the remainder of these are basically all of the big beds, most of which are commercially harvested. Next, please. All right, so ultimately, what we would like to know is what the seed output of each of these beds is. If we're going to model this process, we need to know how many seeds are entering the water at each location along the coast. And the way we get there is sort of a four-step process. We go into the bed and we estimate the density of mussels, and then we measure the area of the bed. Next, please. And once you have those two, you know the population size. So we're essentially doing a little mini sort of stock assessment here. And then if you combine that, the size frequency just means how many of them are what size. And then we look at their egg production as a function of size. And when you put all of that together, you get the reproductive output of each bed. Next, please. And so what I'm going to do is sort of step through these four pieces of the process and tell you a little bit about each one. Next, please. So looking first at density. To assess density, we just, within the mussel bed, randomly assign 3 sub-areas, go into each of those, randomly assign 3 transects, and then collect, randomly assign 4 plots or quadrats on each one. And in each of those, we just go in and count the mussels and measure all of them. That's where most of our data are coming from. Next, please. Bed area, we've played with various ways of estimating bed area. So if you can walk the bed, you can actually just divide it into a bunch of squares and rectangles, and then we just, with long tapes, measure 2 sides of each square or rectangle. Next, please. If you can get up above the bed, this is an example from Pleasant River, where this is a little bit of a bluff. You can sit in one place and basically use surveyor's technique to get the range and bearing to a bunch of landmarks around the edge of the bed, and then reconstruct the area that way. Next, please. And finally, we do use some aerial imaging, both from small planes and drones, and that's often a good way to measure the size of the bed. Next, please. In this small bed, we were able to also walk it using a handheld GPS, and you see that the area estimates are very comparable from those 2 methods. Next, please. And then finally, so when you combine the density and the area to estimate the total population size, here you see data for West River done with 3 different ways of estimating the area, and it all comes out within about 20% smaller patches, tidal falls preserve within 10%, no matter which way we do it. Next, please. All right, so then looking a bit at the size frequency distribution. Next, please. I'm not going to say much about how we do it. It's pretty simple. We just measure the mussels. What I'm showing you here, though, is for a bunch of different size classes within each population, 15 populations. I'm showing you what percent of the population is made up of each size class. And we have, for a lot of these, we have data for 2 different years, 2014 and 2015. And pretty much in all of the populations, you see, as you might expect, the mussels have gotten bigger from 2014 to 2015. Distributions are shifted to the right. The thing that's sort of alarming is that you see in most of these populations, as those mussels are aging and getting bigger, you are not seeing very many new recruits coming into the population. So again, these are pretty recruitment-limited systems, though there are the oddballs, like this Tidal Falls Preserve Site, which gets gobs and gobs of recruitment, but has almost no adult mussels. Next, please. All right, so finally, when we sort of integrate all of this, we get to what we were ultimately interested in. Next, please. And so this is just a table of the areas, the density, number of adults and larvae, minus a digit there. And these are just ranked from the north to the south. And you see most of them are pretty big beds. They're numbering in the tens of millions of adults. Next, please. And this one in Machias Bay, as I said, is really small. And this is actually a subtidal bed right near the intertidal bed, which is a very large area, but not especially high density. What we're ultimately interested in is the larval output, and there's a digit missing there. So those all say either a number times 10 to the 12th or 10 to the 13th, which, if you're not used to that notation, 3.0 times 10 to the 12th is a 3 followed by 12 zeros. That's how many seeds these beds are putting out. Next, please. So another thing that's sort of disturbing for a bunch of these beds, we've been able to measure the reproductive output both in 2014 and in 2015. And in all of those cases, so 2014 in blue, 2015 in red, in all of those cases you see a big decline in reproductive output between 2014 and 2015. And the reason for that is because the density was declining. Again, we didn't have much recruitment in those beds. The mussels were getting older, some of them were getting eaten by predators, some were getting harvested, and there were no young ones growing up to take their place. So we see a consistent decline in reproductive output over those 2 years. Next, please. Finally, the last thing I want to talk about in terms of the work in these beds is the timing of spawning and settlement. And again, we set out to quantify spawning time because we needed it to sort of model this process. But it actually turns out to yield some pretty interesting information just in its own right. Next, please. And so the way we monitor spawning is we go out there and we collect mussels. And all we do is we cut out all the soft tissue in the mussel. And then we weigh the gonad, the reproductive structure, and we weigh the rest of it. And what I'm plotting here is what we call a gonad index, which is just of the percentage of soft tissue, how much of that is attributable to gonad. And we're plotting it through time within, again, each of these 15 populations. So here you see sort of the typical pattern. The gonads grow, and then they suddenly decrease, and it's this sudden decrease that tells you that the animals have started to spawn. These patterns look sort of distressingly complex. Next, please. I've sort of just stuck arrows on there to indicate the approximate spawning time in these different populations. Some populations spawn out rather rapidly, some kind of seem to dribble for a longer period of time. Some of them get their act together, they spawn once, and they make new gonads, and then they spawn again. See, these two populations did that. Interesting, this one, which is right in between the other two, did not spawn a second time. It spawned once and called it quits. We've also got populations like Pleasant River, where it appears they never spawned at all in 2015. That's 31 million mussels that didn't get their act together to make seed that year. One thing you notice, for 2 years of data, it looks like most of these populations seem to spawn pretty close to the same time both years. Next, please. And finally, there's actually more order than you might think to all that noise. spawning time varies by more than 2 months, but there is sort of a southwest to northeast progression. So here, this is the day that spawning began in each of those populations, with the populations just ranked from northeast to southwest. And so you see spawning starts in the southwest and progresses to the northeast, though there's obviously a fair amount of noise around that relationship. Next, please. All right, so where this, I think, starts to really get interesting is when you look at the temporal sequence of spawning and also can compare it to the temporal patterns in settlement. And so that's what we're trying to do here. There's no real data on this slide. What I'm showing you are curves fit to the original gonad index data. And the individual who made this slide is colorblind, so you get to see it all in shades of gray that are maybe more distinct to a colorblind person than they are to the rest of us. But the dark mound is the patterning gonad index for the first spawn in each population. Okay, so the spawn actually occurs on the downside of that curve there. The slightly lighter gray is the second spawn in that population, if there was a second spawn. And at the same time we're monitoring these guys for spawning, we're also monitoring settlement. And so the lightest gray is the temporal pattern in settlement, when it began and sort of when it peaks. Next, please. And so you've got this is your gradient in spawning time that I showed you on the previous slide. And where this gets interesting, next please, is if you look at especially at the early part of the settlement, when we're first starting to get settlers down here in these southern populations, well, that's before most of the northern populations have spawned. Okay? So right there you can conclude that these were not the source of these early season larvae down here. Next, please. So we can essentially take out all of these populations. I should add, we know it takes the seed about 4 to 7 weeks to develop in the water. So we can exclude all of those populations, next, and we're basically left with only that list as a possibilities for where those settlers came from. And you'll notice that what those possible sources are either essentially the same bay or bays that are not too much further to the northeast from the heart bay. It's a little, you can try to adopt the same logic to the peak settlement. You don't get quite as far because there's more possible sources. You can only exclude a smaller percentage of them, but I think this is a really inexpensive way to at least get some information about how the populations are connected. Next, please. And finally, if you go back just a second. The way we presented this, we just plotted the settlement as a proportion of the total that occurred at each site. So it makes you think that settlement is equal in all sites. Next, please. And that's really not true at all. So this is showing you the absolute settlement density. Again, this is north to south. You see it's highly variable. This is that site I said that gets lots of settlement, but it's almost no adults. Next, please. All right, so I've sort of been alluding to this throughout that our goal with a lot of these data is to try to model larval dispersal. And the reason you can basically do this is that larvae are really weak swimmers. They mostly are going to go where the water takes them. The exception is they can control their vertical position a bit, which may get them into water that's moving a little slower, a little faster. But what they can't do is just swim horizontally against the tides. So because of that, you can to some extent just model larval dispersal initially with just a really good coastal circulation model. You know where the larvae goes, you know a lot about if you know where the water goes, you know a lot about where the larvae go. And you can make that a little bit better if you then do a sort of a coupled model and include some larval behavior and growth and survival. Next, please. And so what we've been doing is developing a high-resolution coastal circulation model. The black part of this slide shows you our model domain, and this is what we call the grid for the model. And at every little intersection point within that grid, okay, at every one of those, the model calculates the water speed and direction for 20 different depth layers in the water column. And it does that once per second. Okay? Needless to say, this does not run on my desktop. This requires a lot of computing power. And this whole model is driven by a larger scale Narrowtooth model that essentially establishes the boundary conditions around the edge. And when when we're way inshore here, our model grid points are only about 100 meters apart. So this is the higher resolution model than anybody has ever done for this part of the coast of Maine. Next, please. And so a lot of you are probably skeptical about models, and I share your concerns. We've been doing a lot of work to try to establish how good a job our model does. And there are various things we can do. For example, there are tide gauges, 3 tide gauges in our region, and we can compare how well the model predicts tidal height with the gauge. And so here you see a comparison with the Bar Harbor tide gauge in black and the model output in blue, and you see we're getting the spring neap cycle and everything quite well. We also go out and collect data along all the transects shown in red there, and we measure current velocities and the physical structure of the water column and then compare those to what the model is predicting for those areas. And so up in the upper right-hand corner, you see here a comparison between the model output, which are the blue arrows, and the length of the arrow corresponds to the water speed, and the direction just indicates the direction. And then you see where we've collected data on the surface velocities along this transect. We're showing you the actual data, and you see the model does a pretty good job of up in the bay, the flows are lower and they're mostly down bay. And as we move further down, the flows become stronger and angle more to the southwest as we start to hit that coastal current. Now, we can also look at sort of a cross-section, a slice along that transect, because when we measure the current speed, we actually do it all the way through the water column. So here we're plotting our cross-section data along that transect in top here with the current speed chopped off. These are in meters per second. And then we're showing you below this how what the model predicts along that same transect. And you see that overall, there's a pretty good correspondence there. We've got our lowest flows in the same spot, a little bit higher flows here, and then a gradient towards higher flow offshore. There are problems, there are areas where we have problems. Next, please. And if you look carefully at the bottom, you'll see the problem is that there's some bathymetry here that the model doesn't know exists. And any of you who've spent much time on a boat in eastern Maine probably know that the information on your charts leaves a little bit to be desired. And so this is one of those just sort of missing little bits of bottom. And obviously, the presence of that changes sort of the flow around there. We get some higher velocities right in there that are missing in the model. So where the model screws up, it does so mainly because NOAA doesn't have good enough bottom topography information. Next, please. But overall, it does pretty well. So this is an attempt to show you some of the flow patterns in the model. This is sort of removed, after you remove tidal effects, the tides slosh the water back and forth. But probably the easier way to look at it is what we're going to do is just release particles in different locations and track sort of where they've gone to. Next, please. And so you can do this in 3 dimensions. These are particles released in Machias Bay, and you see how they move. It's all quite spectacular, but it's also a little bit hard to extract detailed information from. Next, please. So what we're going to do is just turn and look from the top and ignore what's happening with depth. And so if we release particles out in the EMCC, in the model, they behave exactly like our drifters did. They just go straight down the shore. They don't mix into these bays. One of them went in and played around at the mouth of Frenchman Bay. That's about it. I should add that the color coding here gives you the number of days since release. And if they're out in the EMCC, they basically leave the system very quickly. They're headed down to port. Next, please. If we release them in a bay, we get a rather different result. These are released in the very top of Chandler Bay, where there's a bed up here. And you see they can spend a long time just within that one bay. If you follow the color coding here, in 2 weeks, some of them haven't gotten out of the bay. Those that escape mostly move to the southwest, and they visit a lot of these neighboring bays where there's beds in the West River and in Pleasant River. And eventually some of them will exit and move on down the coast. They also can move a limited distance to the northeast. So you do see a little bit of this northeastward flow, and that basically happens when a flooding tide sucks them up there. So they did move into Englishman and the mouth of Machias Bay. Next, please. And this is just another example. This is a release from Pleasant. You see them very quickly moving to Harrington and actually getting down into Gouldsboro and then exiting the system. Next, please. All right, so that's where we're at on that. It's just at this point, all those particles are just passive. They're just dots. Okay, so what we're going to be doing is adding biology to the model, looking at adding in sort of growth and survival functions. So the particles will grow, they'll die as they move around as a function of temperature. We're looking at adding some vertical movement behavior, and then ultimately all the field data I was talking about feed into this. Put all the reproductive output of these different populations in to release the number of particles corresponding to the output of each bed, and we release them at the time the population spawn. Next, please. So the last thing I'm going to talk about is what we've been doing to develop a natural tag technique to try to empirically actually trace where these seed are moving. And mussel larvae are way too small to tag. Okay? They're by the time they settle, when they're at their maximum size, they're about 1/3 of a millimeter long. But the beautiful thing is they actually tag themselves. And the way they do this is when any bivalve extracts calcium from the water to make a shell, which is calcium carbonate, it sort of inadvertently captures all the other elements that are in the water. And it turns out that the ratio of those elements varies among bays. And so you can use this sort of geochemical signature to assign larvae back to the bay they came from, to assign new settlers back to the bay they came from. Next, please. And so the way this process works is we use a laser interface to what's called a mass spectrometer that does the elemental analysis. And first we develop a reference map to know what that signal looks like amongst the various bays. And to do that, we rear juveniles in those bays at the right time of the year, and then we just zap the growing edge of their shell to get the elemental composition in that spot. And then the other thing we do is we collect settlers from those bays. And next, please. The beautiful thing about bivalves is that larval shell is retained even after they settle down to the bottom. So if you go up to the very tip of the umbo, there's a little 1/3 of a millimeter long bit of larval shell that was laid down not while the animal was growing where it is now, but when it was first conceived in the bay where it started its life. And so if you take your reference map developed with that, you can then assign the settlers back to their point of location, point of origin. Next. And we use these 9 elements just because they're abundant and easy to assay. Next, please. So this is I took this figure from a publication by some colleagues, which just shows that there is variation in these elements, in the ratio of these elements amongst the various bays in Maine. So here we have just 7 different elements, and the amount of each plotted for 7 different sites. And so you see, one site is really high in zinc, and another has 3 sites are high in strontium. And no single element gives you much ability to assign a larva to a bay, but when you use all of them in conjunction, they actually give you a lot of information. Next, please. And so here we're looking at our own data, and these are just the juveniles that we grew in these bays, okay? And we've assayed all 9 elements now in them, and we're taking all of that information for those 9 elements and sort of crunching it down into this 2-dimensional scatter plot representation, so you can see how the different bays are positioned. The circles indicate essentially the means for each population, and the points show you all the individual mussels. And so the first question we ask is just, okay, we know where these came from because we collected them there. How good a job can the analysis program do of assigning them back to their original, to the bays we took them from, okay? And so with 133 different juveniles out here, it correctly classified 128 of them, and it missed on 5. So that gives it an accuracy of about 96%. That's, I think I would argue, pretty good. It's even more interesting if you look at the actual misclassified assignments, because 4 out of the 5 all involve confusion between Pleasant Bay and Pigeon Hill Bay, which turned out to be kind of similar geochemical signature. And other than that, one paired classification problem, it's really doing a great job on the rest of it. Next, please. All right, so now what we can do is so that's our sort of our reference map, what the signal looks like. We then take settlers from these bays and ask where can we assign them back to. So we tried to do this in 2014 with 9 of these beds, a subset of those that we work with. Next, please. And we only had settlement at 6 out of the 9 sites. Next, please. And when we back assigned things, only 5 of the 9 potential source sites didn't show up in the settlers. Okay, so what we have is information on how larvae that started in these 4 bays ended up distributed amongst those six bays. So not everything we might want, but it's not a bad, it's a reasonable starting point. So starting with larvae that originated in Pleasant Bay here. Next, please. What I'm plotting here are percentages. So of those that originated in Pleasant Bay, 13% of them stayed in Pleasant Bay and settled in the same bed. Next, please. 26% of them moved one bay over to Harrington. Next, please. There was no settlement in Pigeon Hill that year. 48% of them ended up in Dyer Bay. Next, please. And about 12% of them ended up divided between 2 different lobes of Frenchman Bay. So from a scientist's perspective, this is a great pattern. You know, it looks like most of them are moving 4 or 5 bays, and then they're getting diluted out. You don't see their signal much beyond that. Next, please. So we can also look at Pigeon Hill Bay. Seed originating from Pigeon Hill Bay, no settlement there, so no evidence of self-seeding. Here we find that some of them actually did move northeast to Harrington Bay, 25% of them. Only 3% of them showing up in Pleasant Bay. 34% down to Dyer, and about 55% spread amongst the different lobes of Frenchman Bay. Next, please. And finally, for those that originated in this lobe of Frenchman Bay, more than half of them actually stayed in Frenchman Bay, but you find that 16% of them managed to get up here to Dyer, 7% of them made it up to Harrington, and the data are showing 24% of them in Pleasant. And this is the only point of all of these where I really sort of have to wonder whether that's right or not. Next, please. All right, so as I said, the work on the modeling and the natural tagging studies is ongoing. We have to add the biology to the model and do more to sort of validate, to ground truth it. The natural tags, we actually have a very complete data set for all the populations for 2015. We just haven't analyzed it all yet, so I can't tell you what it says. And we obviously, at this point, don't have answers to all the questions. Next, please. However, if I go back to my sort of 3 original possibilities for what's happening with larval exchange, next, please. Everything we're seeing pretty much rules out the supply from Canada idea, okay? The physics as we observe them on the ground with drifters, if there are larvae, we know there are larvae out in that coastal current, but the drifter data says they probably don't mix into the bays. The model particle tracking shows the same thing. The timing of settlement, of spawning and settlement is consistent with much more local sources. Next, please. And on the other hand, we have evidence of both of these other patterns. So the modeling results, the natural tags, and the timing stuff all support both of these possibilities. Some level of self-seeding, some exchange among neighboring bays, mostly moving southwest, but occasionally moving some shorter distances to the northeast. And so what we're really going to be focusing on for the next couple of years is trying to sort out how often these 2 different alternatives occur, and if different alternatives occur more in one bay than in another. Next, please. And I just want to, again, acknowledge all my collaborators and everybody else who's worked on that. Thanks.
We have some time for questions.
Yes?
I watched, as an elk fisherman, I watched the temperature change, and I watched on Maine harbors the sea temperature. I watched the sea temperature on Maine harbors, and it's all color-coded. And it shows every year, the last 3 years I've been watching it, it shows right now, it's showing that the plume of warm water is coming in the Great North Channel from Georgia's, and it's working to the northeast up along. And last spring when we had all the cold water that was in the bay, we watched the warm water push in like this, and it pushed the cold water right back around behind it, but then the warm water came in behind it like that, and it kept the cold water right in the middle of it as the warm water worked in around it. And I'm wondering if your east to west, your west to east settlements are occurring as that Gulf Stream water, the warm water is pushing it to the east instead of the normal northeast to southwest flow that Bigelow back in the 1800s.
So I mean, I think you're basically right. I think there's several things going on. That normal northeast to southwest flow occurs for various reasons, and one of them is, let's say, like wind direction. As you change the wind, you're going to have episodic events where the water goes in different directions, and that can move larvae if they're out there. The other thing is that inside of that coastal current there are eddies. And so something getting caught in an eddy, first off, you've got to understand that on a flood tide, a flood tide, a larva can move 2 bays on one tide. If it's out just a little ways off, out of the bays or on the headlands, it can go that far on one tide. So there's the tides, there's the eddies, and then there's the wind, and some combination. And that all interacts with the temperature structure.
Phil, so how does clam larval duration compare to mussel duration? And so how could you potentially get insights into clam kind of population structure and movement from this mussel study?
Yeah, I don't know all the answers to that, but you know, potentially you can take this approach and apply it to any of the bivalves. Okay, there are reasons we work with mussels, and I'm happy to talk about that. But the big things you're worried about are what time of year do they spawn. In other words, are they in the water at the same time of the year as the mussel larvae? Because everything we do is sort of time specific. And then the other big issue is, what is the larval behavior? What sort of vertical migration behavior do they have? Because if larvae have evolved to try to stay in the bay, all they have to do is drop down on an ebbing tide, because the surface water is going to move out faster than the bottom water, and then come back up on a flooding tide, because the surface water is going to move in faster. So a little bit of vertical movement can make a big difference in terms of whether they get out of the bay or not. And so I don't actually know, and I don't know the difference in PLD. Brian, mussels versus clams, how long do the larvae stay?
28 days, larvae.
Comparable then.
Yeah.
So, you know, but those are the things you want to worry about.
Is there any opportunity to, during the spawning time when they're out there traveling, is there any opportunity to probably place a couple nets at a certain level to perhaps catch some of it?
Yeah.
Transfer in.
Yeah, I mean, I think you could easily, well, the thing I would do is go to that site, Tidal Falls Preserve on the Hancock-Sullivan border, which is a magnet for larvae, and there are no adults there. I would go hang nets there.
But I was thinking of the ones that you have coming from the Canadian side down off some of our bays there to try to catch some of those ones that are moving all the way to Massachusetts and so forth.
Yeah, you could certainly try it. I have tried to deploy things, moorings and stuff out there. It is, anybody here who's worked in the Eastern Maine coastal current will tell you that is a tough place, okay? You ought to see what the lobstermen do to try to keep their traps. I mean, you're talking about an environment where every trap buoy is a polyball this big, because that's the only thing that will stay on the surface in the currents. We've had moorings out there that we would go out there and routinely find them missing, and our standard approach was to zigzag to the southwest for about 2 miles until we found it again. It's really a challenge to keep anything in place out there.
Do you have any theories as to why over the past 10 years, I know on Swans Island, we've just seen a bed completely die. Now, is there anything you could think that would cause this lag in spawning? Is that something that's reciprocal, or?
Well, I think, you know, the long-term decline, I think, is probably the function of a lot of different factors. You know, it's the predation regime has changed, the temperature regime has changed. I don't think you can single out one factor. One thing that I find really interesting is we found that 2015, it wasn't a great settlement year, but it was a better settlement year than 2014. I think most people would agree with that. Yet, all of our measures show that larval production was lower in 2015 than it was in 2014. So that sort of screams that it's something that's happening to the larvae in the water. So I can't tell you exactly what that is.
Under attack.
But it's that step in the process, maybe.
Yeah.
Yeah, I think we're going to move on. Thanks, Phil. Next time you speak, we'll have a disco ball for you. Dr. Beal is going to speak next on spatial variation and cultured soft-shell clam survival and wild clam recruitment in the Harrison River. I'm going to turn the mic on for you.
All right, thank you. All righty, let's see if I can advance this. There we go. So my co-authors include Chad Coffin, Clint Goodenow, Sarah Randall, Kyle Pepperman at PPI, Ben Ellis at PPI, and Cody Jourdain, all at Down East Institute. And I want to say thank you very much for Phil. That was a very impressive and outstanding bit of work and science. And so last year, I talked to you about some results that we obtained in Harrisika, where we set out what I'll call a comparative experiment. We're on both sides of the Harrisika, only within about 2,000 feet of each other. We set out the same experiment, and the design was fairly straightforward. We planted clams in plots that were netted at either 15 or 30 per square feet. We had 10 groups of 4 nets each, and the X's represent places where we actually had put out green crab traps that were fished twice a week, and we removed the green crabs from those spots and asked whether or not we actually needed to fish green crabs as well as net. And the answer to that first question was that the netting the green crab traps didn't contribute much at all in terms of adding or decreasing the survival of the soft-shell clams. So we learned that nets alone are going to be just as effective as nets plus traps. So that was one thing. But the other thing that we learned, and that I told you about, which is why I'm going through these slides fairly quickly here, is that we got completely different results on either side of the river in these 12-foot by 20-foot plots that were netted. This is a picture from November of 2014. We took core samples from each of those 40 plots on one side and 40 plots on the other, so a total of 80 benthic cores. We washed each core through a very fine mesh 1 millimeter sieve, and then we counted the clams and looked at their survival. And at Collins Cove, which is on the western side of the Harrisika, we found very, very high mortality rates. Pathetically high if you're trying to farm clams or grow clams in a clam stock enhancement program. In terms of the number of wild spat that we found under those nets, it turned out that there was about 3.1 clams there in April, and there was about 3.1 clams there in November. So the netting did about as well in terms of protecting the wild clams as it did the hatchery seed, which wasn't a lot. Across the river, however, we got completely different results. Anywhere between 85% to 100% survival, and excellent growth at that. But one of the key factors is that we found, on average, about 1,400 clams per square foot that were wild. So on one side, 3 clams per square foot, and 2,000 feet away under the same kind of nets and in the same kind of environment, mostly very, very gooey mud, we found 1,400 clams per square foot. So the question is, are these kind of results repeatable? And so we just did a little bit of back of the envelope mathematics here and took a 12 by 20 foot plot, said that if we found about 1,400 recruits per square foot, that we ought to get somewhere close to 1/3 of a million clams underneath each net. And when you do that math, you end up with somewhere around 13 million clams that weren't there to begin with, that were a result of doing this particular netting program. All right, so that's where I left off last year. And so what we did this year was to look at both sides of the river. This is approximately the places where we had put out the experiment in 2014. But in 2015, what we said we'd do is to take a look at both sides of the river and create essentially 10 sites that are somewhere between 500 to 600 feet apart, that go all the way from where we're a little bit before where we seeded. This is, whoops, let me go back, there we go. Let's see. So this is the area that we would have seeded last year, and there. And so we started a little bit south of where we started from the previous year, and then worked our way up along this eastern shore, and then again worked our way up along the western shore. And in each one of those sites, what we did is we put out 4 plots. And the plots were about the same size as we used this past year in 2014. We seeded at about 20 per square foot, and this would be what it would have looked like if we had gone in, or when we went in and did the seeding. These would have been the sites at, oh, the cursor isn't showing, okay.
That's Bill's.
That's Bill's. Well, then that means that I have to be able to know how to use this. There we go. Okay, thank you, Bill. So these are the plots that were at Collins Cove, netted, seeded with about 20 clams per square foot. And this is what the design would have looked like at each one of those 10 sites on one side and 10 sites on the other. And just a small schematic that would have, this is what it would have looked like from the air. Okay. So this is what we're going to get for percent survival of clams on the eastern side, and what we can see is tons of zeros, and the best that we were able to do is about 25%. Many of the clams came back dead and undamaged, so when we planted the clams, they didn't go anywhere from underneath the nets, they stayed there, but we found an awful lot of dead and undamaged clams. And we found a lot of dead and chipped clams. Well, you know that clam can't get chipped by itself. Something's got to do it, and obviously it's got to be a crab or some other crustacean. How can clams be dead with undamaged valves? I'll explain that in just a moment. But here are the averages. Less than 5% survival underneath those nets on that western side. And 53% were dead and undamaged, and about 40% were dead and crushed under netting. Okay? This is what we got on the eastern side. And there's a few zeros, and there's a few areas that are greater than 25%. But still, I have to say it's nothing that I really want to write home about. In fact, it's not really as good as the year before. We still had plenty of dead and undamaged clams, and we certainly had plenty of dead and crushed clams, once again, under netting. And so if we look at those averages, there's about 25% survival, not 85%. 28% were dead and undamaged, and 47%, almost 50% were dead and crushed. That's not a very good result if you're either trying to enhance stocks or if you're farming. So what's going on? Well, this is what's going on. These are milky ribbon worms, and these are responsible for the deaths of these hatchery clams. The milky ribbon worms, it turns out, are kind of like clammers. They wait for clams to get to be a certain size, and then they go in and they eat them. They're not really interested in the very small clams. They're interested in clams that they get to be about 35 to 45 millimeters. 35 millimeters is about 1.25 inches, and 45 is almost 1 3/4 inches. So they're waiting. They're waiting for these clams to get to be a size, and the nets aren't doing a thing to keep them out because they're burrowing infaunally. They're in the mud, and they rarely come up to the surface. They just go from one piece of mud to the other. Now, what we also found out by doing our core sampling was the density of these worms. And they're very high. 3 per meter square doesn't sound like a lot, but these are worms that get to be about this size or about this size. And so when you put 3 or 4 of those in a square meter, the biomass of these worms is great. And 3 on one side, 4 on the other. Well, what about green crabs? We haven't said a lot about green crabs here at the Fisherman's Forum since really 2013. But I want to tell you that during 2014 and 2015, we continued to trap green crabs at most of the sites that I've X'd up there on the east side and west side of the Harrisseaget. There's still plenty of green crabs around. They're mostly small, but the numbers are almost as high as they were in the years where, they decimated clam populations. And so we estimate from, again, the cores, these are just simply crabs that are in the mud inside these nets. Well, there's 3 or 3.5 per square meter. And, that's enough to do some damage underneath those nets. So crabs and milky ribbon worms are certainly explaining a lot of the mortality that we see there. We found very few clams that didn't grow before they died. Now, if you look at these clams, they have this unique ring that is shown that occurs about the time that they're seeded. It's a disturbance line, and it occurs in all the seed that we've planted from DEI and from before then, the Beals Island Regional Shellfish Hatchery. And the size that they're planted on is recorded in there, which is why we don't have to mark these clams, because like Phil's clams, but not elementally, these are marking themselves through disturbance. The other problem that happened along the way is this, and the best adjective that I can use to describe it is bullshit. And this is a small gastropod that doesn't exist in eastern Maine. I didn't see it really until I started this work in Freeport. It's the mud snail, Ilionassa obsoleta, and it weighs down these nets by laying its eggs on the nets themselves. And we've tried a bunch of different techniques, brushing them, using high pressure water. There's a guy that wants to lick them off, but that doesn't quite work. But anything that we've tried to do doesn't seem to work. So we ended up having to replace these nets because they got weighed down so much that if we hadn't done so, the sheer weight of the netting would have made everything under the net anoxic. And in fact, in some places, the nets did go anoxic. So some of the mortality that was undamaged clams was probably due to the results of the weight of these eggs. At the end of the day, what we were able to do then is to go in and take samples like we had done before. And we were also able to look at small clams, wild recruits. And if you take a look at the number of wild recruits that are occurring on either the west side or the east side, and you look at the number per square feet, you're certainly not seeing anything close to 1,300, 1,400 clams per square foot under those nets. So 2015 was different from 2014. But wait. I want to talk a little bit about clam reproduction, and this kind of gets to some of the work that Phil was doing, although not in the detail that he was. So a little bit about clam spawning, and let's see, can we get this video going? Does that video work? Maybe it does. Okay, so clams, like mussels and like scallops, are broadcast spawners. You can't see it very well, but there's the siphon of the clam. And all of these are chunks of eggs that are coming out and are mixing with and being fertilized by sperm. The eggs are about 50/1000th of 1 millimeter. Okay, and so that's about 1/125th of an inch. They're very, very small. But imagine, and these are clams that are being spawned purposefully at DEI. But you can imagine that this is happening in the mudflats, all up and down the coast. So that's what's happening. Those eggs are being distributed into the water column where they're fertilized. And moments after the sperm has fertilized the egg is not going to be round anymore. It's going to be this oval shape, and now it's going to start dividing, and it will continue to divide and grow, and the cells will differentiate, and some of them will be producing gut, some of them will be producing cilia or hairs. Anyway, they're going to end up developing into a swimming larva, and that swimming larva looks like this at about 10 days old. This is an animal that has a shell, and this is that larval shell that Phil was talking about in the previous talk that gets laid down about the time when when they begin laying that shell and incorporating calcium carbonate and the other elements that are available in that particular water column. We know, and Phil's going to be looking at this in future, but there's this question about, if you look to the east, no matter where you are on the coast of Maine, is that where your larvae are coming from, or are they coming from the embayment? And so his work, although specific to mussels, Chris, I think will help us understand a little bit more about soft-shell clam and where they go. So this is a 10-day-old larvae, and these slides, by the way, are being taken by, these videos are being taken by Kyle Peppermint. Kyle, can you stand up for just a second, please? Thank you very much. Kyle Peppermint, who used an iPhone under the microscope to take these photos with, which is pretty cool. Anyway, Phil said that settling mussels are a third of a millimeter, settling soft-shell clams are a fifth of a millimeter, or about 200 microns. And so they're out there looking for substrate. This is what they look like when they come out of the water column after their 21 to 28 days swimming. And this is what they're going to look like when they settle into the mud flats or onto the sand flats or wherever. Notice that they have a foot. They're looking for something to burrow into. All right, so it's this spawning, this larval life cycle, this larval development, and then the settlement to the benthos, or to the bottom. And so I was very interested, and I call this the salt shaker effect. I've talked about this before. And I envision it as that the larvae are just coming from the water column and they're settling routinely and maybe randomly along the mudflat surface. So at the same sites that we set out the 4 net plots at along the Harrisicot, we put out boxes, wooden boxes. And the boxes are 1 foot wide and 2 feet long, and they're either top and bottom with PET screening. It's like a window screen, but it's pretty heavy duty. The aperture is about 2 millimeters. It's fairly small. And then we use a material called Vexar, which is a much more rugged material. It is a 1/4 of an inch, or 6.4 millimeters, versus about a 1/12 of a millimeter. So this one had PET screen on the top and the bottom. This one had Vexar on the top and the bottom. And I figured that this screening would keep out milky ribbon worms from coming up from below, and perhaps from coming in from the top. And I figured this one wouldn't, because milky ribbon worms can go through a 4.2 millimeter mesh, or a 1/6 mesh, so they ought to be able to get through a 6.4 or a 1/4 inch mesh. I also thought that green crabs might get into these boxes, because I know that green crabs are in the 4 to 5 to 6 millimeter size range in terms of their carapace width in the fall. And these were put out in April on the mudflats. So I envisioned that what I was going to be able to see was something of recruitment. And you saw the recruitment under the nets at those sites was abysmal compared to the year before. So what we did was we put out 6 of these boxes at each of those 10 sites on each side of the river. So 120 boxes went out around the middle of April. Two of the PET screen boxes I added 1 liter of play sand to, thinking that they might need some substrate to settle onto. The boxes are full of air. The boxes have no clams in them at all. And they're anchored on each end using some stakes that are hooked to a piece of heading twine that's on each end of the box. And we're using a lath with a notch carved in it, or sawed in it, to catch the becket, or to catch the handle here. So they're anchored to the mudflat surface, and there they're going to stay from April until November. The idea is to take advantage of the salt shaker effect, to see whether or not these boxes would collect larvae. And if they collected larvae, how many would be collected in the Vexar boxes versus the PET screen boxes, but also how many would be collected in one part of the river versus the other, or one side of the river to the other. April 11th is when we set out most of the boxes, not all. It took a few tides to do it. And this is what they look like in November. We hauled this box up and turned it over, and look, there's a milky ribbon worm that was on the outside of it. It had been there. It had been on the underside, but didn't get in. Out of the 120 boxes, we found no milky ribbon worms in any of them, even the Vexar boxes. But what we did do was find out that almost every box filled completely with mud, which we thought would happen. The mud is in the water column and just settled out, so that was very interesting. We took each box and we took the Vexar or the PET screen off, and we took all of the mud out by hand and put it into a 1 millimeter sieve and sprayed the contents of each box through a 1 millimeter sieve. And then we counted everything that we could count, and there was an awful lot to count. In fact, here's one box that our good friend Kyle actually sieved that had 6,009 clams. Again, there was air in this box in April. And by November, it had 6,000 clams, or about 3,500 per square foot. Here's a box that had 52. So there was a lot of variation. 109 of the 120 boxes actually had clams. So there was only a few zeros. Here's a box that had 1,800 or so, or 1,600 clams. But not only did they have softshell clams, they had a clam that, as an adult, gets to be about not even quite a half an inch, called Neolinia lateralis, or the dwarf surf clam. There were false angel wing clams, there were razor clams, there were tons of mussels in some of these. So as Phil said, a lot of mussels growing on mud flats, and yep, they can see to mud flats as long as there's some kind of substrate that they could attach to, and they attach to the inside of some of these boxes. This is what 1,600 clams looks like from one of the boxes. And you can see that there's a whole variety of different sizes, and that was something that really surprised us a lot. And more about that. So here's the average number of wild clam recruits per box, and you can see that here's the scale over here, and most of the sites are less than 50 per square foot. Really nothing really to write home about. But like I found the previous year, there was a fairly high settlement in the area where we actually had seen the 1,400 clams per square foot the year before. There was actually more than 500 on average per square foot in that one site. So there looks to be some kind of a pattern that's consistent from one year to the next. That's the first really consistent result. In terms of the number per box, yes, there were fewer in the Vexar, but there was not significantly fewer. They averaged anywhere between 14.5 to 23 per square foot. On the other side of the river, we found that the PET screen boxes had more than the Vexar, and in both instances, there weren't any more clams in the ones that we put sand in originally than versus just having no sand at all, just air. In addition to measuring 20 clams at random from each box, we took the smallest clam and the largest clam from each box and measured it. And so the smallest clams ranged from about 2 millimeters all the way up to about 15 millimeters, or anywhere between 1/12 of an inch to a little bit larger than 1/2 an inch. That wasn't surprising, but what was surprising was how big the largest clams in the box were. Now remember, these clams are not crawling in there from the mud, they're settling in there from the water. And there weren't any clams in any of the boxes to begin with, yet at the end of November, some of the clams were as large as almost 1.5 inches. That's incredible growth, and I had no idea going into a winter that a clam that's what I call a 0-year class clam, or a first-year clam, a clam that's only a few months old, could possibly be that large. When we look at the shells of clams, we see these lines, and we use these lines to try and age the clams the best that we can. And this is going to force me to age clams a little bit differently. The average size of these clams, where was that? The average size of these clams was 22 millimeters. 22 millimeters. So that was the small and the large. Let's just take a look at the east side versus the west side in terms of sizes. So these were among the 20 randomly measured clams from the boxes on the west side versus the east side. You can see that the clams on the west side were a little bit larger than on average than the clams on the east side, but still a wide variety and wide range of sizes, which means that there isn't, like Phil just showed us from eastern Maine, there's a very wide range probably of when clams are spawning, and certainly when clams are settling. Now, what I'm trying to say here is that I don't believe those clams are the same age as these clams here, and I don't believe these clams are the same age as these clams here. So we're seeing an early settlement, and then we're seeing dribble settlement all the way through the summer, all the way down into the late, maybe the early fall. The first couple of years that I did this, where we didn't use any of these boxes, we found a lot of small clams, 7, 6, 5 millimeters going into the winter. Those obviously had to settle very late in the year, and I think this data shows that that's the case, since the early settlers are fairly large. But where are those early settlers under normal conditions? Well, I think they become food for a lot of those predators that are out there, because we rarely see 35, 36, 37 millimeter clams going into the fall. In terms of the PET screening versus the Vexar, the clams in the PET screen were actually a little bit larger on average than those in the Vexar, probably because the ones in the Vexar may have gotten eaten more by green crabs, since we did find more green crabs in the Vexar. But in terms of growth rates of crustaceans, this study also gives us a little insight on how fast green crabs can grow. Now, you know that crabs and lobsters don't have a way like soft-shell clams and mussels to incorporate their growth into their shell. So it's very difficult to age these things. But I think that this is a very good technique for learning how fast these crustaceans grow. This is a 45 millimeter, a 46 millimeter green crab that was found in this particular box that had 0 clams in it. 46 millimeters. Okay, it's in a box with Vexar. So if you do the Pythagorean theorem, a 6.4 millimeter mesh is 9.05 on the diagonal. So I'm thinking that, you know, if you tip the crab one way and allow it to go into the box, this crab, I think, was a minimum size of about maybe 9 to 10 millimeters in April when it entered the box just by random chance alone, and then it just stayed, and it molted, and it became trapped. And then it probably ate all of the settlers, whether they were mussels or bloodworms we found in these things, dwarf surf clams, etc. But that's phenomenal growth. I don't think that that's been recorded. Again, that one had no live clams. And this is the size frequency distribution of the green crabs, all the way from crabs that recently settled, recent to November, in the 2 to 3 millimeter size range, but also green crabs as large as that. But we found green crabs on both sides that were pretty much the same. The average size, as you can see, is 17 and 18 millimeters respectively for each of the sides. And in terms of PET screen versus Vexar, a few of the larger ones we found in the Vexar, but still we found green crabs as great as 30 millimeters in those PET screen boxes, and I think those walked in as 2 millimeter animals or 2.5 millimeter animals in the spring and became entrapped and grew to 30 millimeters over that time frame. I'm not even going to bother with that. But so the summary is, you know, we got started on this to try to look at attempts on a large scale to see what we could do to enhance stocks in Freeport. And everything that we've done to date, and I hope to be reporting something different next year this time, but everything that we've done to date has been marginally or completely thwarted by predators, whether those predators are invasive or whether those predators are actually common or endemic. And so This has led to a lot of inconsistencies, both spatially and temporally. However, I think that results that I haven't been able to present here, but that I will in another time. But I think what the results show here is that large-scale attempts, probably not, but small-scale attempts on the size of an acre or 3 acres, I think are going to work. And I think that there are ways to keep out these predators so that somebody can make something to go except, on the scale of 1 to 3 acres. Some of the patterns that we see for recruitment were consistent from one year to the next, so that was pretty interesting. And then growth of both first-year clams and green crabs, I'm saying must be reconsidered in light of these results. And then finally, clam reproduction, and certainly clam recruitment is certainly not a single event, but appears to occur over the course of 2 to 3 months in the late spring and the early summer. So I want to thank everybody that helped me in the field, Mike and Jim and all those folks from Freeport, and these are the folks that contributed to the funding for this particular project from NOAA, CPAC, CNET, UMM, Maine Community Foundation, and the Down East Institute. And I said to Phil, I'm going to end on the slide you started. This is DEI. Thank you very much.
Great, so Dr. Beal can take a question or two, and then he's going to be around for the networking session as well.
Yes.
Any initial observations or thoughts about the difference, west side versus east side? I know there's a lot more human population on the west side there.
All I have been told about the hydrography of the river is that a dye study was done to look at the effluent from the sewer treatment plant that's on the western side. And the dye study showed that there was a faster current on that western side than on the eastern side. And so I don't know if that faster current is sweeping larvae off or and allowing them more to settle on that side. At this point, I don't know. So I'll have to go back really and take a look at those hydrographic studies and see if that's the case. Dana?
You have a speculation as to, on the size range, how much of that might be original settlement versus vessel drifting into the canyons and starting at a little bit larger size? Would that be possible?
Well, there's not a lot of vessel drifting from Maya. There's a lot of vessel drifting from Midless. I still have the samples. I didn't do much with we got some mussels that were almost 2 inches in the Vexar boxes, and so that's probably vessel drifting. But in terms of Maya, no, there's not that vessel drifting. Yes, Dan?
The boxes netted top and bottom.
The boxes were netted top and bottom.
So did you have to fill those, then place them into the mud?
No. Well, I put them down as empty boxes, and I walked away. And the mud that got in them was mud that was in the river that just simply over time, just settled out. The height, they were made out of strapping, so they were about maybe 1 by 3. Exactly. Yes.
Yes, on the clam boxes that had both clams and green crabs,
Yep.
what was the survivability rate of the clams?
That's a great question, and it depended upon the size of the crab. That one that had 6,009 clams in it also had a green crab.
Really?
But the green crab was less than the size of my thumbnail. So it made a difference as to the biomass completely of the crab. When the crabs got to be around 1 inch to 1.25 inches, there was no live clams. Anything less than that, and there were some live clams.
The observation is key.
Yeah. Chris?
So these growth rates are unbelievable. I mean, they're just huge growth rates.
Yeah.
And so, because like, we just did some Sharpie marking and got about 6 millimeters, and you've gotten similar kinds of rates of growth for a season. Is the fact that these guys are up in the water column a little bit, just a tiny bit, right? Could they be getting different food or anything like that just because they're, I mean, it's not very much different. I'm just trying to, these are pretty mind-blowing numbers.
Well, that's a great question. I don't know, because I don't have, well, Are they similar to what growth rates that I've observed with clams in the flats and protected? Yes.
Okay.
At these sites.
Okay.
But even in the one that had 6,000, the largest clam in there was 35 millimeters. So, you know. One more question?
Yeah, I think you had these two guys.
Okay. Yes.
The milky ribbon worm,
Yeah.
it waits till the clams get to a certain size.
That's what we've found, yes.
It doesn't leave the bigger ones alone or anything, it doesn't.
Well, it concentrates on the larger clams.
How does it actually do in a clam?
How does it do in a clam? It's got a wonderful proboscis that it diverts, and it can either go in through the siphonal holes, or it can go through the pedal hole, and it diverts, it emits a toxin that kind of makes the clam tissue become all liquefied and just, you know, lovely if you're a worm. They just kind of go in and snarf it out. They leave no shell damage at all, so, yeah. Yes?
Did you experiment leaving both screen sides exposed to the water? So in other words, tipping up one of the boxes on its side?
Yeah, no, it didn't. All the boxes were laid down flat because I didn't know how to anchor them the other way.
Oh, yeah.
Thank you.
Okay, our schedule sort of slid a little bit, so we may cut into the networking time, and I just want to warn everybody in advance that if people are cool with this, we're probably just going to kind of skip the break. People can go to the bathroom when they want to. And we'll just move into the networking as soon as we can. This room has got to be freed up right at 4 o'clock because it's used for the reception. But you guys can go and just network in the hall if they kick us out, so we can just continue the time that way. The next session we have is on microplastics, and our first speaker is Ivy Kanyoka. I hope that's close. She's the new baykeeper for the Friends of Casco Bay, and then she's going to be followed by Abigail Barrows. You want to change the mic?
Good afternoon. Can everyone hear me? So my name is Ivy Franyoka, and I'm the new Casco Baykeeper with Friends of Casco Bay. I started in January. And for those of you who are not familiar with the Waterkeepers movement, there are 270 waterkeepers around the world. And our job is to advocate for and protect and restore the water quality of a particular body of water. My body of water is Casco Bay, and I work for Friends of Casco Bay, which has a team of scientists, researchers, and educators who all work together to protect Casco Bay. And we're very happy to be here talking with you today. And the issue that I'm here to talk about is, what do white teeth, smooth skin, and our seafood have in common? And the answer is microbeads. So what are microbeads? Well, according to the federal law that will be regulating them, they are tiny plastic spheres that are less than 5 millimeters in size. They can be made of many different types of plastics, and as you can see, they can be many different colors. Microbeads are most commonly found in a lot of common cosmetic or facial cleaning products, body scrub products, and whitening toothpastes. And this gives you an example of the size of them. You've probably seen it if you have kids like I do who use like clear silk facial scrubs or whatever. This is what they look like. And this just shows you how small they are. So why am I here talking to you about microbeads and microplastics? Because they get into the ocean and they affect seafood. So there's really 3 main sources of microplastics. One is the cosmetic products that we were just talking about. The other, which I was really disappointed to learn about, is from our fleece jackets and other synthetic clothing. As it gets washed, the fibers wash off and are too small to be trapped, and those plastic fibers enter the water stream. And the other is plastic litter that, as it breaks down, can break down into really small particles. And most of these small particles of plastics are so small that they just come right out the end of sewage treatment plants. There are very few sewage treatment plants, there are none that I know of in Maine that have the level of screening that can filter out microplastics. And they end up in the ocean. So how do they impact ocean life? You're going to hear a lot more about that from my co-presenter, Abby Barrows, who will be up here shortly. But just generally, they affect the growth rate of marine algae. They're absorbed by marine invertebrates like sea cucumbers and worms, and they're ingested by shellfish, and they impact the digestion of the shellfish and get absorbed into the intestinal walls. And they can also cross over into the circulatory system and cause higher respiration rates. And so if they're in organisms that are either directly food for humans or in a food chain for humans, then the next question is, how do they affect humans? And there has been a little bit of research done in this area. For example, one European study found that an average portion of mussels has about 90 particles. So someone who is an average consumer of shellfish would eat about 11,000 microplastic particles a year. And there was just recently, I just saw the study published yesterday, a study from California where they have tertiary treatment on sewage treatment plants. So the primary source of plastics that they found was the particles from fleece. And there they went to a seafood market and measured the amount of plastics in fish actually for sale, and they found that at least 25% of shellfish samples had microfibers in it, and 67% of the fish. So what are the impacts on humans who ingest shellfish with microbeads? We don't really know yet. These are areas that are just beginning to be studied. So at Friends of Casco Bay, that means that we are talking with other researchers such as Abby and other baykeepers around the country who are doing this research. And we're designing our protocol and our pilot projects so we can do more field tests to determine how much plastic is out in the bay and the impacts of it. And in the meantime, we know that plastics is a problem, and people have long known that plastics are a problem for marine life. So we need to take steps to eliminate the amount of plastic that's making it to the ocean, because it's so hard to deal with it in sewage treatment plants. So some of the things we can all do in the meantime is those little string-like things in the corner are like microfibers, and we can support companies that are beginning to do research to try to change the size of the fibers so that they no longer break off and go into our sewage treatment plants. There are also companies that are trying to develop filters for washing machines. We can reduce our use of plastic by using reusable bags, bringing utensils with us instead of using plasticware, those kind of things. We can download this app, it's free, and you can take this with you to stores and you can scan barcodes and it will tell you if a product contains microbeads, if you still want smooth skin. There are plenty of products out there that contain natural exfoliants that break down and don't harm marine life. And the biggest thing we can do is support laws that reduce or eliminate the use of plastics. And Maine did this last year. Maine passed a law banning microplastics. And Maine was one of 8 states that passed such laws. And because states were starting to do it, it made something happen at the federal level. So in December of 2015, President Obama signed into law a ban on microbeads in rinse-off cosmetics. And this law is a really great first step. It prohibits the manufacture and sale of rinse-off cosmetics that contain plastic microbeads. And it takes the place of any state law that's already been passed in this area. And One of the reasons why it's good that it takes the place of the state laws that have already been passed is because 7 of the 8 states that passed laws had this exception saying, well, if it's a biodegradable microbead, then you can still use it in cosmetic products, but there really aren't any biodegradable microbeads, so this closes that loophole. On the downside of this law, it just deals with rinse-off cosmetics, so there are many other products that contain microbeads. And this is how the new federal law will work. By July of 2017, companies can no longer manufacture rinse-off cosmetics. And rinse-off cosmetics, like we talked about, are face scrubs and toothpaste and those kind of things. By July of 2018, they can't sell those products anymore. And they also can't manufacture cosmetics that are also drugs. So a cosmetic that is also a drug might be a facial scrub that has something added to it to treat acne or to reduce the appearance of wrinkles. And then by 2019, they cannot sell those rinse-off cosmetics that are also drugs, like the acne scrubs and the wrinkle products. So this deals with cosmetic products, but it doesn't deal with a lot of other products that contain microbeads. And that's something that we'll also be looking at is as more data is collected off the coast of Maine and we begin to have a better idea of where the sources are of these microplastics and what types of microplastics there are, then we know maybe what kind of legislative initiatives or regulations are needed to address this issue. And of course, the reason we want to do this is because, like you, we think shellfish and seafood is delicious, and we want it to be around for all of us to eat. And I am going to, I'd be happy to answer any questions about our work and about this topic, but due to time, Abby and I agreed we do our presentations back to back, and then we'll take questions.
Hello, my name is Abby Barrows, and I'm a marine research scientist based in Sturgeon, Maine. But I wear a lot of hats, so I have worked for the Marine and Environmental Research Institute, and that is what I'll be talking about today, the research I did while working for them. They're based in Blue Hill. I also am the principal investigator on a global microplastics project for the company, a nonprofit in Montana. And I'm an owner of a sea farm called Longcoat Sea Farm in Deer Isle, and I'm a graduate student at College of the Atlantic. And just a little bit more about my background with microplastics research. I've looked at potential sources of microplastic pollution in the Gulf of Maine. I've looked at microplastic distribution throughout the water column. I'm the PI on a global microplastics project looking at microplastics in surface water and microplastics at depth in the world's oceans and freshwater systems. Conducted a comparison study looking at 2 different field techniques commonly used to sample microplastics. And today I'll be talking about microplastic ingestion in commercial seafood species. So Ivy did just talk quite a bit about what microplastics are, but for those of you who weren't in the room, microplastics are, can you guys hear me? Microplastics are pieces of plastic that are less than 5 millimeters in size, so that's about half your pinky fingernail. They're non-biodegradable. They might break down into smaller and smaller pieces, but they never fully go away. They've been shown to absorb and release toxins. They're made from oil, and everywhere that researchers have looked, they have found microplastics. So In all of the world's oceans, in remote alpine lakes, in the deep sea floor, there's plastic. So plastics are typically broken down into two categories, microplastics. We have the primary microplastics that Ivy talked about, which are the nurdles, so these plastic pellets, which are really the feedstock for creating all the plastic items that we use. They're melted down, chemicals are added to them, and they are a fork or other items that we usually just throw right away after we use them. And these nurdles are shipped all over the world in shipping containers, and that's why we often find them on our beaches. And then the microbeads, which are the personal care products. Secondary microplastics are larger pieces of plastic, such as bottles and bags, that break down into smaller pieces through mechanical and chemical degradation. And the one that Ivy just spoke about, which is really upsetting, is synthetic clothing. So every time you wash your polar fleece from Alpine or your Gore-Tex jacket, it can shed up to 2,000 fibers of plastic, which, as Ivy said, is not captured currently with our wastewater treatment plant systems. The filters just aren't small enough to capture these fibers. So even if you're on a home system, your septic tank is pumped out and sprayed in some field, and that all makes its way to the ocean eventually. So why should we care? Not only is it gross to have plastics all through our oceans and waterways, but there are a lot of really nasty chemicals associated with plastics. So in the marine environment, there are floating around all these legacy compounds, DDT, phthalates. These chemicals are hydrophobic. They don't bind to water molecules. And they are lipophitic, so that means they're fat-loving, and they are attracted to plastics. And all these chemicals have very nasty known health effects. Also, plastics are made, 50% of plastics are made with hazardous materials and chemical byproducts, which are also then released into the environment which they're in. So ingestion, it's scientists have been finding lethal amounts of plastic in marine mammals and fish. And as we look closer, we're also finding microplastics. This isn't surprising since global annual production is over 300 million tons of plastic as of last year, and about 12 tons of plastic are thought to enter our world, 12 million tons are thought to enter the world's oceans each year. So that's about 8 tons of plastic a minute, to the equivalent of a garbage truck full of plastic entering our ocean. So there's a lot of plastic out there. And unfortunately, it's not going away. It might be breaking down into smaller pieces, but it's still there and affecting wildlife. It's estimated that by 2015, the weight of plastic in the ocean will surpass the weight of fish in the ocean. So here's just a schematic to show how microplastics can move through our food chain. So the lower trophic levels ingest plastic. Larger animals eat them, accumulating more and more plastic as you move up through the food chain and ultimately to humans. And microplastics, as I mentioned, are not just on the surface of the ocean, they're all through the water column and very heavily in our sediments. So it's really affecting all of these different animals. Here's just 2 examples of studies done looking at microplastics transfer in our food chain. You're all familiar with the blue mussel and the green crab. The blue mussel was seeded with plastic and then fed to green crabs. And it showed that, yes, the plastics remained in the gut of the green crab. Some of it passed out. But what I thought was really interesting in that study is that this picture here is showing the gill of a green crab. And all those little flecks are pieces of plastic in the gill. So when there's plastic in the water column, it's not just being ingested, but it's sort of permeating throughout the body of these animals. Here's another study that is more relevant to Maine. Whiting fish were put in a tank with 2-year-old polypropylene fishing line, nicely weathered. Those fibers shed into the water, were consumed by the whiting, and then fed to the Norwegian lobster. And lo and behold, when you open up the lobster, many days later, there are still plastics in their gut. So some of the effects, I'm not going to go through all of these, but basically these studies have all shown that ingested microplastics are really negatively impacting our marine species, from increased mortality, endocrine disruption, transrelocation to cellular pathways in blue mussels. That was a study done in Nova Scotia. So this spelling pretty bad news in terms of the health of our marine species as they ingest microplastics. So this led me to want to do a study, albeit tentatively, to look at microplastic ingestion in some of our favorite Gulf and Maine species. So at Mary in 2014, we collected mussels, both rope grown and wild, oysters, clams, lobsters, and mackerel. And in our lab, we dissected the lobsters and mackerel and took out the digestion tract. And for the bivalves, we took the whole animal, and we did a digestion process in the lab to break down the tissues and look for plastics. This is where we sourced the seafood. So my hometown of Stonington, I gathered the majority of the seafood. I got the oysters from the Baggadoos River in Brooksville. Sorry, I have a chest cold and I'm losing my voice. So the results were really upsetting, I thought. Not very scientific word, but I was pretty blown away by our findings. 96% of the seafood samples, we had 51 animals in all, analyzed contained microplastics. They averaged 2.5 microplastics per gram of tissue across all the species. And interestingly, the lobsters and the mackerel, the larger animals, had more larger size class microplastics than the bivalves. So the 3.5 to 5 millimeter size plastics were more common in the larger animals, whereas the bivalves had more smaller pieces of plastic, so showing that they're filtering out these smaller pieces of plastic from the water column. So on the graph here, W stands for wild mussel, RG stands for rope-grown aquaculture mussel. On the y-axis is the average number of microplastics, and of course the animals on the X. I want to note that I thought it was interesting that the wild mussels had more microplastics than the aquaculture mussels, and I attribute that, this is just a theory, that the wild-caught mussels I collected off Stonington Harbor, they're old, big old mussels. And so they had a much longer time to sequester microplastics versus the younger rope-grown mussels. So this is the numbers of plastics. So we have the average total per organism, and as you can see, oysters had the largest amount, and the wild-caught had more than the aquaculture. But when you look at the average pieces per gram of tissue per organism, the wild mussel had less microplastics per gram of tissue, and that's because the mussels were much larger than the rope-grown ones. We also broke the lobster down into tamale and then its GI tract, and I was surprised, just another reason not to eat tamale, even though it was this least amount of plastic in the animal, there was still plastic present. And once again, the mackerel had the largest amount of microplastics across all the species, which when you think about the food chain, it's not too surprising because the mackerel is eating a lot of the smaller animals that might have already ingested microplastics, therefore it has more of an opportunity to sequester those plastics. So future research directions that I think are really important in this emerging field are to determine the residence time of plastics in the guts of these animals. How long are they staying in there? There's been a lot of lab studies, but it's also important to look at wild species. Are they past flushing a lot of these plastics? Are they in there for 20, 30 days, years? No one really knows yet. Also to quantify the toxins that are being transferred from microplastics into the tissue of the animal, which is very difficult to do because they live in a semi-toxic environment with a large opportunity to interact with some of those persistent organic pollutants I talked about. But that is really important to understand that toxic transfer throughout the food chain and how that affects humans who eat seafood. I think that it's really important to use this sort of data to leverage legislation on plastic reduction. Because without letting these studies get out to the public, we're not going to be able to implement changes in our daily lives, in the state, and across the nation. And ultimately, we all want to be able to eat healthy seafood and also commercially fish healthy and sustainable seafood. Thank you.
We have time for a couple of questions, if people have questions for Abby or Ivy. You guys want to come up and answer some questions?
You mentioned sort of a maximum size on a piece of, microplastic. What's the smallest end that you can observe? Would the little fibers from a fleece jacket have registered in that study?
So in the seafood study, we used 0.8 micron filters to filter the digested animal. So potentially down to 0.8 microns in size, but for that study, we were finding around 1 millimeter size for the seafood bivalve.
I happen to notice that you didn't have any of the numbers on soft shell clams.
I was hoping someone would notice that, and it's very unfortunate since one, I really enjoy clams, and I know a lot of you guys are clammers. The clams were the first animal that we processed in the lab, and we had a lot of problems with the glassware breaking as we heated them, so we had to throw out our samples. But the initial, I think we looked at 3 out of the 10, and they all contained a healthy amount of plastics, or an unhealthy amount of plastics, we would say.
Do you think the lipid content of the species has more of an effect in terms of either they're absorbing or retaining the plastics?
I'm not sure if the lipid content would affect the plastic ingestion, but I think it would definitely play a large part in the amount of chemicals that would be leached out of the plastic and into the animal. Fatty tissues attract toxins, and if that plastic is traveling through the animal and it's a fatty animal, then yes.
Our town has a different type sewer system than most other towns. Our town is served by sandhills, and I don't know if you're familiar with sandhills, but they have approximately 4 feet of a certain grade of sand from the top to the bottom, and all the effluent goes through the sand before it actually gets to our plant where it's treated. Have you looked at any of those type systems to see how much, if, I would assume that they would track more of it than a regular treatment facility?
I personally haven't had a chance to do a lot of sampling inside of wastewater treatment plants. I've done tours of the Stonington plant and the Blue Hill treatment plant. But other studies in Europe and in California show that the different types of wastewater treatment plants do retain different levels of plastic, although it doesn't seem like there's any one that is the surefire one that is really helping straighten out the microfibers.
Yes, just going to add that is an excellent question, and it's one of the research questions that has to be looked into. In New York and New Jersey, when they looked at the harbor they studied the sewage treatment plants, and of 33 plants, I think there were 26 that had microplastics coming out. And so the question, I asked the same question you did, why did it not come out of the remaining other plants? And in that situation, I think it was because they had that highest level of treatment, tertiary, but nobody knows for sure. And so I'm really glad you asked that question, because it's something that we should, as researchers, be looking at to understand if that makes a difference.
How long is this study going to go before you release it publicly? And
So
how
the-
expansive is your testing going to be?
So this was really just a case pilot study that we completed last year. I'm working on a methods paper right now, but this is next in line to be published. And I'd like to publish it and be able to release it so anyone can access the study.
Abby, could you tell anything about the origin of the microplastics in the mussels or clams or tamale?
That's the question everyone wants to know, so they can either point a finger or feel like it wasn't clear. It is almost impossible to trace the plastics back to their origin, other than sampling in a wastewater treatment plant, because so many of the plastics are similar in terms of where the polymers are similar polymers, so it's very hard to point a finger at one place in particular.
I think to add to that, one of the, some of the other studies, what they've done is in looking at the types of plastics that they're finding is, or in doing these kind of studies, to look at the types of plastics they're finding so that they can tell if it's more foam or from styrofoam containers and things like that, so that then they have a sense that it's coming from takeout coffee cups and large containers and things like that. And that has not been done yet in Maine, and I know it's probably one of the questions that we're looking at. Should we be doing a catalog of the types of plastic fibers that we find?
That would be very helpful, I'm sure.
Also, in a study just came out last week showing that the plastics that they're finding through surface trawls in the Atlantic are much older plastics than the plastics they're finding in the Pacific. So they can do some aging of the plastics, but you can't tell exactly where they're aging.
I know there's no going back. Has anybody ever looked at breaking down the vinyl coating on the wire lobster traps and polypropylene line that's been used for too long?
That one study I showed with the whiting and the Norwegian lobster, they were using aged polypropylene line that had been used, I assume, in the fisheries. But people aren't necessarily focusing on the vinyl coating breakdown. One positive thing, although they did use it out in your yard in the winter, is that at the bottom of the ocean is colder and much more stable, and so the breaking down process is much more slow compared to the surface plastics.
Have you noticed differences in the levels between towns with overboard discharge plants, like what's his name had mentioned, in towns with privately owned septic systems?
That's a good question. I've definitely spent some time looking at those maps, but we're just finding so many plastics everywhere. It's pretty hard to say that this town has more because of X, Y, or Z.
What's the state of engagement of wastewater treatment professionals these days? Do you care about this, Phil?
The tours I've gotten, people are really interested in it, but the question everyone has is how could we possibly have a filter small enough that would be so energy intensive, or something that would have to be changed every 20 minutes to capture these fibers. So it's definitely an engineering question. I think there's interest. I have found, for the most part, all the wastewater treatment people really lovely to work with and really willing to show you their facilities and discuss ideas about how things would be implemented in a more effective way.
Yeah, I would agree with that. I asked that same question based on the studies I saw in California, New York, and New Jersey, and it is very cost prohibitive to look at doing this at the treatment plant level. So it would be interesting to find out and really understand better why some treatment plants currently right now are screening those out well. And I don't think anybody's really addressed that question because I know I've asked it. And then the better solution, the more economic and sensible solution is just to be eliminating some of these sources in the first place. Like, especially if we can use something like sea salt instead of microplastics in a body scrub, then it makes sense to make those kind of changes.
What would your end game be with this? Like, say we got it legislated so all of these plastics that are created in fibers were no longer. If you're telling us already that it's through the water column and in every body of water that you've tested, and we can't get every other country in the world to do the same thing, what is the end game here? How can we keep our fisheries from having this problem if short of filtering the entire world, what would the end game be?
That, of course, is a really good question, and that is the kind of question that, that's why we're designing these pilot studies, because part of what, it's kind of like how we deal with ocean acidification. If we can't stop carbon emissions from all over the world, what can we do in our coastal area? And we know that a lot of these sources of microplastics are local. So if we can eliminate those and maintain the high quality of that Maine seafood is known for by having less plastics or eliminating them as best we can, then I think we just have to be realistic. Like I couldn't do what I do. You guys couldn't do what you do if we weren't optimists at some level. So there's these threats out there, and all the questions that have been asked are ones that I want to take back and think about when we're designing our pilot projects, so we can get answers to the good questions that you have and try to make a policy that means that we have high quality shellfish and seafood.
Thank you, everyone.
Thank you very much. So we're going to go on to our last presentation of the day, and this is on some water quality success stories, I guess you could call them, or just water quality stories, partnerships. But you have to read them. It's all on there. Okay, so our first speaker is Melissa Smith, who works with the Department of Marine Resources Bureau of Public Health.
Hello, everyone. Everyone hear me well? So, we're in Togo. No, Everfield. So my job was Phil Young was my graduate advisor when I was doing my master's degree, and his hypothesis that Canadian exports head straight to Massachusetts is indeed not totally correct because I settled in Maine. I have been here for almost, 12 years, 9 years with the Department of Marine Resources. This is my third year working with public health. And the talk that I developed today is almost the work that I have not participated in, except for the last 2 years. So this was sort of a history lesson for myself to get me up to speed on some of the areas that are within my purview as a growing areas manager. And there are 4 growing area managers in the state of Maine, and I cover 1/4 of the state. I take care of Kennebec River and all rivers between Damariscotta and Stockton Springs. So the premise of the session today that ultimately Allison had put together was the concept that water quality and bacterial pollution is something that we take care of at DMR, but it's not something that we do in a bubble. It's definitely not something that we do by ourselves. And without having collaborative partnerships, we would never get to where we are in some of our growing areas to have more harvestable acres, shorter closure times, shorter day length of closures. And so I just want to go through today and sort of discuss how it is that we go about looking at a focused area and having the partnerships that we have to do a total analysis of the area and to hopefully have an end result of improving water quality health, but also to increase harvesting. So the National Shellfish Sanitation Program is ultimately the guidelines that we follow at the Bureau of Public Health. And it's the FDA that gives us the model ordinance, which is where we sort of have a baseline of the minimum rules and regulations that are set for us. The state can go above beyond those, but we must at least have the baseline rules set. And in doing that, the area classifications that come from that model ordinance are dependent on us either finding or isolating point and non-point source pollution. And I realize that saying that we're isolating non-point source pollution is sort of an oxymoron, but if it's not a septic system that's malfunctioning, or if it's not a wastewater treatment plant, but it's a cove that is heavily inundated with wildlife year after year, those are, that's an example of a non-point source solution that we can identify and document. Other things that we need to do area classifications are updated shoreline survey records and, of course, water quality. So the program essentially runs on risk management and pollution. All areas that we have within our purview right now, the management polygons are put in place based on risk management. What are the levels of bacterial pollution that are present in those areas, and how can we put in place and enforce a management plan that would reduce the risk of public health getting sick from eating contaminated shellfish? And in order to do this, we go through and we have focused sample collections to identify adversities. Adversities could be a season, it could be a marina, it could be an overboard discharge system, it could be a wastewater treatment plant, it could be river flow rates, it could also be something that includes all of those things all at once. And so having areas that are impacted by multiple adversities is, I suppose, the biggest hurdle that we face as a department, because it's very difficult to isolate out how those adversities interact with each other, but how do we manage when they don't always interact at the same level or intensity. Our management plans are then refined based on the data analysis that we collect. And when we add extra data to our already existing database, we are then able to either shorten closure length or reduce area size of a management zone, therefore increasing harvest rates. So for today, we wanted to highlight 3 different collaborative research projects that have occurred within the Department of Marine Resources over the last several years with other partnerships. And today there's only going to be discussions on the Kennebec River and the Dominic River primarily, and I have like 1 slide for Richmond today. And so I'm going to take you through the history of the Kennebec River, and then the next speaker will discuss the Dominic for you. And the Kennebec River is in my purview for my growing area, but most of the research that has been done was before I started working with the Bureau of Public Health. So it was really interesting for me to go back into our history and to see what has been done and how long it's taken us to get to where we are today, because these studies are definitely not quick. This is a view of Atkins Bay that I had taken, I guess, 3 weeks ago with a colleague. We were sampling. And that would lead me into the history of the Kennebec River. So prior to 2008, the Kennebec River estuary had different conditional management plans that were either rainfall or season, or a combination of rainfall and season. For example, Todd Bay was seasonal, whereas Atkins Bay was rainfall adverse. And then, current management as of right now, all of the Kennebec River is managed based on river flow discharge rates. And so we use two gauges, one in the Androscoggin, one in the Upper Kennebec. We take the combined river flow rates from those two USGS gauges, add it together, and then that number dictates how we manage the river based on season. It was in 2001 that the FDA did an investigation into the Kennebec River estuary that, with the results of that study, concluded that the estuary itself, during high river flow events, also had high fecal coliform pollution. And so, that having come out in 2001, we didn't put our first river flow management plan into play until 2009. Since 2009, we've been adding data and refining that management plan to get to what we have, what you see up on the wall right now, with 4 different zones and 3 different seasons of river flow discharge rates. So how did we get to where we are today? By many, many partnerships. In 2009, one of the big questions that was happening after we first implemented the river discharge rates into the Kennebec River is that 50% of harvest time was cut when river discharge of 30,000 CFS was implemented. And once the gauges would combine for CFS, it was an automatic 14-day closure. And so with that, we didn't lose harvest space, but we lost harvest time. And so local shellfish groups, local land trust groups all got together with the question of what can we do? What can we look at that would assist in increasing harvest time for everybody within the Kennebec River system? And so in 2009, this Kennebec Estuary Partners Coalition was formed, and they actually found a 2009 Clam Day poster that they had made. And I'm just going to read off some of the list of partners here. So it was several local conservation groups, so the Kennebec Estuary Land Trust, the Sheepscot Valley Conservation Association. There was multiple state agencies, both DMR and DEP, and there was 4 different soil and water conservation districts, Androscoggin, Lincolnville, Knox, and Waldo counties. On top of that, there was 6 different townships involved, Aroostook, Bath, Georgetown, Pittsburgh, Westport, and Woolwich. And then, on top of that, there was also all of the available Kennebec watershed shellfish committees. That's a huge partnership to get together to look at the river system, but that's what was needed. And the work that started in 2009 also included AmeriCorps volunteers for both DMR and CALT. And in 2010, that's when DMR began collecting adverse data at different levels of river flow within the Kennebec River. So once the partnership was formed, there was a couple different phases of monitoring work that was done by DMR, while the other partnerships were doing their part in assessing water pollution, doing shoreline surveys, doing stream work and stream enhancement, doing workshops within the town that would educate homeowners on domestic septic systems and maintenance and how to keep them working. And while that was happening sort of outside of DMR, within our walls, we were having volunteers from the community, from shellfish industry, collecting additional data, which resulted in phase 1 in 2010, having the results of water collection at different flow rates allowed for us to implement a management plan in 2012 that gave us 3 different levels of triggers. So no longer were we only managing at 30,000 CFS, but we were able to have seasons that during the winter, we were able to have a 60,000 K CFS, and in the summer, a 40,000 K. So that allowed for less closures and more harvesting. Phase 2 study happened in 2012 between DMR and several volunteers from both Pittsburgh and Georgetown townships and shellfish committees, in which a very intensive sampling program occurred that they would harvest on days 1, 3, 4, 5, and 7, both collecting meats and waters. The data from the 2012 was analyzed in 2013, and that's when I finally got to see what was happening, what was being done in the Kennebec River. And the results from the 2012 intensive study resulted in a 2014 management plan that built upon the seasons of trigger, but with the extra data from the meats and the water study together, we were able to reduce the closure time. So instead of having a 14-day automatic, we went down to having a 5, 8, or 10-day, depending on the season. We were also able to eliminate the seasonal closures, and we could eliminate the rainfall closures. So by 2014, the work that was done included additional data on our part, but also a lot of stream work from the AmeriCorps volunteers, a lot of shoreline survey work from community volunteers, and all of that together helped us identify what the pollution sources were, both point and non-point, and to give us a better idea of how to manage those pollution sources that were coming down from upriver to the lower river. So now this is 2016, and there are still several pollution items that are still on that original list that was first sort of talked about and discussed in 2009. And so moving forward from here, we want to tackle a phase 3 research project of upriver effluents. And so currently right now there are point and non-point pollution sources present in both the Kennebec and Androscoggin watersheds. And we want to be able to assess the impacts of these multiple wastewater discharges that occur on both of these rivers to see how they affect lower river, Atkins Bay, for example, in the winter and fall. Does chlorination year-round benefit the lower river? So with this, we now have new partnerships. We are teaming up with, again, Department of Environmental Protection, DEP, who are now doing E. coli testing bi-weekly for 1 year on 8 of the upstream wastewater treatment facilities. These 8 municipal facilities are north of Meading Bay, and so in combination with what DEP is sampling up above, we are also sampling down below. And so we are doing a sort of a concurrent study, I suppose, that when there is a high flow and a 1-inch rainfall trigger, we send our staff to go out and sample these stations that you see listed on the side. And what we're trying to do is determine whether or not can we detect non-chlorination. How far down does the potential non-chlorination occur? Do CSO events impact the lower river? And so this is an ongoing 1-year special study that we're going to complete. And as of right now, we are managing based on all the information that we know, and it is our goal, it might be lofty, but the more information that we have, it's possible that we might be able to decrease the size of the prohibited area in Drummore Bay. We know that if any event is happening upriver that does not extend down to where the divide is right now between the prohibited zone and the conditional zone, and we can prove that there's no adverse effects during high flow events or rainfall high trigger events, then it's possible that we could shorten the prohibited zone and therefore opening up more harvestable lake. And so those are sort of the goals that we have while doing these collaborative research projects. So this is a photo that I stole from the internet from Friday Grieving, who works with the NEST program. And this is Frenchman's Bay. And I just wanted to highlight one thing about Frenchman's Bay is that the DEP also have this program called SWAT testing, and it's surface water ambient toxins. And when we do these water studies to determine the influence of pollution into a certain bay or estuary or inlet, having additional information can help us determine what the management plan is going to be. And so having SWOT testing done is just another tool in the toolbox that we use when we have a list of partnerships going forward and trying to get new areas open for harvesting. So just wanted to circle on back to one of the first slides. Our goal is to manage risk and pollution. And primarily when we're dealing with conditional management of areas, we know that there's an adversity that's affecting those waters. And that adversity can be, point, being a water treatment facility, or it can be non-point. It could be land runoff, it could be rainfall impact, it could be seasonal. And any of our research projects try to either isolate or further refine what it is those pollution points are and how we can better manage for them. And with that, I'm going to leave Phil to talk about the mighty Menominee.
Phil Garwood from the Department of Environmental Protection is going to speak next.
As she said, I'm Phil Garwood from the Department of Environmental Protection. My daytime job is to do enforcement in our water quality division, which primarily involves working with wastewater treatment plants. Every now and then, I get a chance to do something fun, like what I'm about to tell you about. Hit the down arrow, nothing happens. Okay. Oh, okay. It doesn't show on here. All right. So, the subject of today's talk is the Menominee River, which is in Waldoboro, Maine. If you look in the insert, you can see, if for anyone who might not know, Waldoboro is located in the mid-coast, and the area that we're really interested in is right at the bottom of the highlighted arrow area. That's the growing area. But the watershed extends up about 32 miles above tidewater. And about halfway up, you can see a little blue triangle, which is Menominee Pond. That was the extent of any of the surveying work and sampling work we did during this project. And this is a blow-up showing Waldoboro and the conditional area within the growing area. Waldoboro, the Route 1 is the diagonal line right at the top edge of the picture. And then the village area starts there and goes down. The red is the prohibited area. And then the blue area, the dark blue area, is the area that's conditional. And the conditional area is very important. The background for the study is that clam harvesting is a major part of the local economy. It can be over $1 million. In fact, I think this year it's closing in very close to $2 million just in the landings value of the clams. And that doesn't say anything about the economic value from the shellfish dealers and restaurants and everything else that's involved. The most productive part of the Menominee estuary is the conditional area. And it's conditional on rainfall. So when there's an inch or more of rainfall in 24 hours, there's a 9-day closure. Melissa had mentioned the meats and water study. There was a meats and water study done about the same time as the Menominee by DMR, and that was used to reduce the standard 14-day closure to 9 days. But even with the 9-day closure, in a rainy year, half the days available, potentially available for shellfish harvesting can be eliminated by the rainfall closure, and that's a huge economic impact. So in 2012, after having worked with several other partners and locally, DMR and the Shellfish Committee in Waldoboro were both very concerned about not being able to make progress. So they pushed really hard on a number of state agencies in the town to form a collaborative group. And the collaborative group included basically people from all of these co-authors plus a few others. The reason I don't have everyone who was involved in the collaboration is that I'm focusing today really on the water quality work that we did, bacteria sampling, and a little bit of related work. So there's myself, two other folks from DEP. Melissa Evers is a freshwater bacteria expert. Becky Schaffner is a GIS expert. And then Melissa Smith and Paul Canova were both very involved in either the design or the sampling or both. Two of the fellows on the Shellfish Committee were very heavily involved, Glenn Melvin and Abden Simmons. Glenn Melvin, in particular, was an Iron Man in this project. He was involved in almost every effort that we made. And then the Waldoboro Utility District was involved. John Fancy is the superintendent. Waldoboro used to have a discharge into the harbor area, into the prohibited area, but they now have a land spraying system where the wastewater is put on the surface of the ground about 8 river miles up from tidewater. And then the Menominee Valley Land Trust was very influential in this. Liz Petruska was the executive director. She's moved on, but we had a large number of volunteers that were brought together by the Land Trust to help with sampling. So the overall project involved water sampling, sanitary survey work, which is mostly by myself and the town, outreach by the agriculture part of DACF, which is Agriculture, Conservation, and Forestry. Waldoboro Utility District, anytime there was a situation that involved a sewer part of the village, the Waldoboro Utility District got involved. And in addition to volunteers, the Land Trust did a lot of the public outreach type work. So in 2013, DMR, as many of you probably know, every shellfish area that is going to stay open has to be sampled at least 6 times a year. And they look for 30 samples over a 5-year period to assess the area. Well, in 2013, DMR tried to double their effort, but because of rainfall, we're able to get about 3 extra. And at the same time, volunteers sampled in a number of locations, a number of stations in the village area, and upstream of Route 1 all the way up to Mid-Otter Pond. Because some of the earlier work had indicated that there were some sorts of pollution problems coming down the Menominee River that were affecting the shellfish area. The other aspect of the study was rain event sampling, and the volunteers sampled on days 1 and 3, and DMR sampled days 5 and 7. Every time, well, we tried to get 2 incidents each season where the rainfall had been above the 1 inch in 24-hour trigger. And DEP, a large part of it was myself, but Melissa Evers did a lot of upstream sampling. Mine was spot checking, and she was doing a lot of freshwater work upstream. 2013, about 2 miles or so above Route 1, there's an area called Winslow's Mills in Waldoboro. There's a seaweed processing plant there. And the worst hot spot we found in terms of bacteria concentrations during 2013 was where Cross Street crosses Menominee River, which is where the kelp plant is located. We determined that the kelp plant was discharging huge concentrations of bacteria, but it wasn't from their internal process. What was happening is water was coming from the river with bacteria in it that they were using for cooling water, and their condenser had cracked. And so the water that was being condensed from the sewage that didn't have any bacteria on it, in it, was harboring massive growth of the bacteria that had already been in the river, 3 or 4 orders of magnitude, and then discharging it back into the river. We found a few other problems, nothing huge. There were some hot spots occasionally, but that was really the only place that was consistently hot. And then there was one area in town where we had frequent high numbers, but not always. Upstream of that Winslow's Mill site, there really were no high numbers. There was one farm that had been bad years before, but the numbers had already improved and continued to improve during the study. But we're still seeing elevated scores during rainfall. So even after we clean things up, we're still seeing the rain effect. And 2013 was a fairly rainy year, so that, I think, played a part in the bacterial results. As a result of that data, I'm not even going to try to show you the data set because it won't even fit on an 11.5 by 22, or whatever it is, 11.5 by 22 piece of paper that you can read without a magnifying glass. Huge number of data points. But DMR concluded that the rainfall closure was justified, and in fact, the numbers looked bad enough in 2013 that they were concerned that they might have to downgrade the river even beyond conditional to restricted. In terms of data, this is jumping ahead just a little bit, but one of the things that we were concerned, that the group was concerned about, was we had found this really high source coming down the river, and was there any chance that that was involved in raising the numbers in the boundary? If you look at this, the 3 sites, WSO 46, WSO 43.9, and 41, 46 is actually a freshwater station that's just below Route 1. 43.9 is at the town landing, which is pretty close to the head of tide. And then 41 is at the boundary between the closed area and the prohibited area and the conditional area. So what we saw, when you looked at the normal 30 sample data sets, which are 8 to 5 years, the numbers had all been fairly high. The geomean near at the boundary of the conditional area was only slightly elevated. At the end of 2013, at the end of 2014, many of the numbers looked worse. We didn't have 30 samples after the repairs were done at the kelp plant. We only had 3 or 4 by the end of 2013, so this data set goes to the end of 2014. And if you see the pre-repair number, we used 11 to have comparable numbers. The pre-repair numbers are really bad all the way across. So that chunk of the data, that 11 sample chunk of the data, was the worst part of the data set. And what it appears is that in 2009, there was a lot of clean data that fell off the data set when we moved to the 5-year window down a set like 1 year. And so that when you look at the post-repair data set, that's considerably better than everywhere else, except for there's a high number at the town landing. And there's something else going on at the town landing, and that'll become evident in a few minutes. In 2014, at the suggestion of DMR, we brought in an environmental consultant, FB Environmental, that works with a canine unit to try to detect human bacteria sources. And they did a dry weather study in the village area, and then once we got the results from the canine study, there were some things that didn't make sense to us, so a subgroup got together and designed a 2-part study for the village area. One of them sampling dry weather at 8 main stem sites, and then a wet weather study with enough, bracketing the main stem, but also including some of the side tributaries in the village area. So for canine detection, they've got dogs that are trained, just like drug sniffing dogs or bond sniffing dogs, that when you present them with a source of human bacteria, they will alert. This dog barks and is rewarded by getting his ball, because he's very ball-centered. The other dog sits down when it detects bacteria. So what we did, what the process is you take a clean hands sample where you have gloves on and a plastic bag, you take a water sample and you put that in the bag to make sure that it doesn't contact any aspect of a person when it goes in the bag. And at the same time we took a bacteria sample. And then you go to a neutral site and you take the water sample and you put it on the ground and you walk the dog by it. And if the dog alerts, it means the dog is detecting human sewage in that sample. If there's no alert, then the dog's nose is saying there's either no human or it's so low that it can't detect it. They will also walk drainages and look for sources, whether it's a pipe or a malfunction from a septic system or whatever, on the spot. And this is a really busy table. The only thing I want you to really focus on is the colored column, about 3 in from my side, and the 2 next to it. The thing that confused us, or that was really surprising about the dog study, was that the fecal coliform values were the 3 highest ones that we had. The dogs said there was no human bacteria involved in those sites, in those samples. 4 out of the top 5, there was no human. The only one was like the one that was 58, I think. And 58 really isn't all that high. It's above standard, but if you're going to dilute that into tidewater, it might not even cause a problem. So that was really confusing to us. And the other thing that was really surprising is that a number of the sites that were well within the open standard for shellfish harvesting, the dogs said that there was human bacteria involved in those sites. So it was really confusing to us. So the village area, Route 1 is up at the left, and at the bottom you can see the tidewater station. This is from our GIS project, and both the size and the color of the dot indicates the bacteria concentration, roughly. The red ones are very high numbers, and the green ones are clean numbers. So during dry weather, I'll go to the table. On the left is the dry weather data. We had moderate levels, low moderate levels in the river main stem sites, except for the landing, which was always within the standards. We had one little blip, so we're not sure if that's an artifact or if that's just a small slug that's coming down the river. But even though the values were a little bit higher in the main stem area, we weren't seeing that at the landing. One of the things we did at the landing was we made sure we were on the outgoing tide about halfway through the tide to minimize any effects of eddies or upcurrent flow. In the rainfall study on the right side, we sampled, it was almost not even day 1, it was almost day 0. And we already had high numbers in all the main stem sites, and particularly in the tributary stream. But those weren't being seen at the landing. And the next day, these were 3 days in a row, the next day, either that slug had already moved through the system and we didn't see it the next day, or it wasn't reaching the landing. So and the landing never really showed, bad numbers. I mean, they were above standard, but not dramatically above standard. So because of that, we designed a study this year, in 2015, to look at all the streams that are discharging directly into tidewater, all the small streams. And DMR stations are the magenta ones up the middle channel, which were new, and then the green ones, which are their standard monitoring stations. And the triangles, whether they're blue or yellow, are samples that the volunteers sampled at high tide, because the only way we could do this project was at high tide for access reasons. We couldn't get to all those sample sites in an adequate amount of time. So another pretty busy table. But on the right here, you can see the scale, greater than 1600, which is higher than DMR, the limited DMR's normal test. And then 2 moderate ones, the lighter oranges. The 32 and the 163 are geo-mean standards for DMR, and then below 32 are, the better stations. A couple of things. about the fifth one down, I just used the one letter in there, Q. It's NME 00, U0Q00, was high for 3 of the dates. June is the left 2, August is the middle 2, and end of October is the right 2. But we had 1.5 inches of rain, 1.25 inches, and then 2 inches of rain, or very close to 2 inches of rain. We couldn't confirm all year that one station was bad because it was underwater. We couldn't even sample it in October. So that one's going to need more study. But the thing that we really are trying to focus on is places where we're consistently seeing bad numbers versus areas where we're consistently seeing good numbers. Because we're looking for smoking guns, and we're also looking to find out what streams we don't have to worry about anymore. And so you can see with the yellow and the green, there's a significant number of streams that are probably not causing the problem. And then we had that one problem. These are all, I should have said, these are all on the east side of the river. And there are 3 sites right next to each other, most of the way down in the October set. I went out and surveyed those areas after we got those high numbers, and the only conclusion is that it's got to be birds, turkeys and geese, because there was nothing else there that could have been. The middle set, the August set, we think that was either an artifact due to stagnant water, or it was a small quantity short-term pulse, because we didn't see that in the estuary data. So, and bear in mind, this is water coming from streams, freshwater streams. This is the west side. The only real issue on the west side, there may be a little bit going on down in the upper part of the conditional area. The only real issue was the sample, second from the top, called J. K is right at the landing, J is the next stream down from the landing, and those two are both fairly high. We're seeing a lot of high samples at the landing, and Melissa even had some very recent data that was showing some kind of a problem that appears to be coming from the vicinity of that sample site. And this is the DMR data. The yellow is the new stations, and then the 3 dates. You can see that there's some elevation on day 3 and day 5. In the spring, most of the conditional area was good by then. In August, the only place we're seeing anything even remotely approaching an elevation is right at the landing. The whole rest of the conditional area was clean, so that's why we think that either that was a very low volume storm, even though it was 1.25 inches of rain, or it was maybe an artifact due to stagnant water because we had so little water to try to sample. And then in the October storm, it was a much bigger storm, and we're seeing that the higher bacteria levels persisted for a longer time. So we've already done some follow-up work. This is the vicinity of the sample site called J. I sampled the rainstorm after the one we sampled there, later in November. The numbers above Pine Street, now bear in mind, this is all sewer. These houses were all on sewer. So the 3 numbers, they're not great up above the road, but they get worse the closer you get to Tidewater. So it indicates that there's something happening between the road and the river, and we don't know yet. We even went out with the sewer district and smoke tested that sewer. Smoke coming out of all the vent pipes in all the houses, but none coming out of any holes or cracks in the ground or the sewer system. So we want to study that further with a dye test this spring, but we really don't have an answer. There's something going on in this area. So status, we have improved water quality, but not enough. It's still reactive to rainfall. We can narrow our focus. We know, we've got 5 or 6 streams that look like they're at least heavily involved in the problem. But because of the dog study, we know that some of the high bacteria numbers we've got are not coming from people. They're coming from dogs, birds, cows. We haven't identified that. So future directions, microbial service traffic. One of the problems we have is we don't have the money. That's a very expensive proposition to do an adequate level of microbial source tracking where you're identifying, okay, it's this species or it's whatever. We are gonna do more spot surveying, and I know that there's at least one grant application been filed, submitted to work on the Madonic. I don't know the status of that. It was put in association with Nest. And then I know the town was working on a non-point source grant application, and I don't know where they are with that process. So I'd be happy to answer any questions.
Do you have any questions for Phil or Melissa?
Thank you, Phil and Melissa. I don't know if it came across, but that was an enormous amount of work, both of those projects from a bunch of different groups, and I think it's really the only way to answer some of these complicated water quality questions. To improve shellfish access and